Is there any virus inside? That's the question one. Question two, what kind of virus that you're going to see inside? Right. And the third one, how are you going to get the virus from the sample? And then you can grow in the lab. So these are actually the basic questions when someone send you virus sample, in the lab. Other than detections of the nucleic acid, PCR, that one is just to detect the nucleic acid. But me, I want to I want to know the virus. I must get the virus growing so that I can test the virus. Okay. You get the PCR, you get only positive for COVID, but you cannot do any testing after that. If I receive a sample containing virus, I get the virus, I grow the virus, I can do many things with the virus. So that is the idea. So, sterile technique is virology. You need to have you also need to know when you handle the virus, you also must make sure that you are not infected by the virus. Am I right? You handle the virus, people send you samples. Sample can get, I mean, they from any labs, from animals. It can be dangerous to you. So sterile technique means that you also need to be protected from those viruses. So in virology lab, there are two ways: security, safety against potential infection to you, and also contamination in the sample. There are two things. So meaning that, first of all, you need in virology lab, what you need to wear? Lab coat, right?
Yes.
You know lab coat. But where do you keep your lab coat? Now, you tell me. You go to many lab, physiology, bacteriology, you use the same lab coat. Where do you keep your lab coat? In your room, right? Your room, or in the office, or in the what we call this, in the lab? Where? In principle, the lab coat being utilized in that particular lab shouldn't be transferred to another lab. But you as a student is okay. It eventually cannot. Lab coat to protect yourself. It is not what we call this a lifestyle as a student, to wear lab coat, no, to protect yourself. But if you take your lab coat from physiology, virology, next week to the what we call this parasite, and then after that bacteriology, then after that to postmortem. Can you imagine that your lab coat, going anywhere, and what kind of thing that you carry with your lab coat? Think about that. That's number one. In principle, cannot do that. Every time you need to wash, you need to actually, in principle, if you want to be very cautious, we are to claim, but we don't do that. Lab coat. Oh, your hands. Don't simply touch anything in the lab. Because no, imagine that virology lab, bacteriology lab, from where the sample? From anywhere. From where? What kind of places that you need to be careful with the lab? Benches, instrument, the tool inside that pipette they use. The water tap. Every time you go, you open the tap water, then actually your pen already maybe carry something. You have to be cautious with that. And what is the most what we call this the place that you have to be very cautious actually? The door knob. Every time you want to enter the door, what happen? You have to you have to hold the door knob, right? And door knobs know how people that holding that knob. Can be anybody. Salesman, lab technician, students, other lecturers, no. If they want to enter to the lab door, they have to open. They have to hold the door knob. So these are usually the potential things that you have to be cautious about. It's you yourself you need to protect. Okay. So some of them wear, certain virus, dangerous virus, we wear gloves, we wear mask. Not the mask that you're wearing for COVID. That one is rubbish, not effective. You see a lot of people going on the street wearing that mask. The pore inside the mask itself actually is very big. Any virus can go through. It doesn't stop. To me, it doesn't stop the virus. It may minimize. It can block mycoplasma. It can block maybe fungus, maybe some kind of bacteria, but virus no way. As long as the wind can go through, the virus go. Maybe the speed is not as high as without mask. Okay. And then these are the things that you need to do about yourself. Okay. Be careful with that. Always wash your hand. Don't make up, holding your hair in the lab, whatever. No, that's not place. Okay. Just be careful with that. The next thing is actually lots of utensils inside the lab. We sterilize them. Scalpel blade, forceps. Okay. How do you sterilize samples? You can never sterilize sample. But what we need to do is actually remember. Lucky the virus is actually smaller than bacteria. So if you have a solution that containing virus, maybe contaminated with bacteria, what are you going to do? We do the filtration. We filter this fluid. Those, what we call these particles smaller than bacteria, can go through. Big bacteria and bigger will stuck inside. So that's why we have a filter, microfilter at 0.22 micrometer. Virus can go through, but bacteria actually 0.3 micrometer and above. They will stuck there. So you actually filter out bacteria. You have the what we call this. You have the sample containing virus. So filtration in the virology is important. Filter, because sample, you destroy the sample, you break the cell, so that the virus come out, will be released from the cell. The virus is the solution, the sterile solution. Then you filter the solution, you get the virus. That's how we work normally. Live virus inside, but very few particles. 1, 2, 3, 4, 5, 10 is not significant. You have to grow them in the cell later on. Later on, you have to take that sample, grow in the cell, grow in other animals. Okay. What about this? The media. We need to use a media to grow the cells. The cell that we want to use to grow virus. We need the media, growth media to grow the cells. Okay. You next particular class you learn that. So you need to do, in order to sterilize, you also use filter. What are the other? You use filtration is actually to separate virus from bacteria. But you want to kill, let's say, this microorganism. What kind of thing that you need? First of all, you about the use of the detergent, right? Clorox. We use Clorox and so on to wash. That will kill bacteria and viruses. Okay, that's what this is actually for the floor, and then when we do that, we use that. But we cannot use it to add Clorox into media to grow cell die. Second, we also do use for the room normally. Room that we work in virology. What kind of thing that we use in the room? We use UV light, ultraviolet light, to kill virus in the small chamber, in the what we call this, the room, a small room. One that we work in cell culture, we use UV light to kill. Okay, but UV light is not effective for the virus in the air. Is very effective, but and when you have scratches on the benches, if you have bacteria inside that the scratch inside then UV light is not effective. You can use alcohol, 30% alcohol or 70% alcohol. 70% can be done, but that's a bit expensive. For a virology lab, 30% alcohol is good enough already to kill most bacteria. Okay, most bacteria. Because alcohol can kill bacteria virus. Alcohol is water soluble, right? So if you use alcohol, it causes dryness. It will cause evaporations of water inside the bacteria and the virus. So the bacteria and virus will exclude. Okay. So that's it. You know autoclave? Have you heard about autoclave? Autoclave. AUTOCLAVE. Then you will learn later. When you visit the lab, you know what it's autoclave about. Normally, when you cook, the temperature, maximum temperature can go to 100 degrees Celsius, right? Steam. But when you use autoclave, they add pressure. They add pressure is high. The pressure is higher than the atmosphere. So the boiling temperature will increase 120, 120. So that will destroy most bacteria. Most bacteria. Virus sure to die. So autoclave. So we use autoclave, you use UV light, you use antigen, we use filtration system. So what else that you do? Okay. In the media that we use to separate, let's say, virus and bacteria, we also add antibiotic, certain antibiotic to that media, so that to prevent potential replication or multiplications of the what we call this bacteria. Yeah. So this is actually important for you to know. So this is actually basic sterile or aseptic technique in virology to make sure that your samples, your place, your utensils, your bottles, your media, whatever, not contaminated by unwanted microorganisms. So that's the basics, including yourself. So don't you see the sample like that and you inhale. You don't know, you may get dangerous bacteria into your body. One of the potential dangerous bacteria is the Pseudomonas from soil. From soil. So soil contains lots of Pseudomonas. So spores actually. Then it goes to your lungs, and your lungs turns green, becomes what? Hulk. Incredible Hulk maybe. Become green now. Then green because of that. It's not because of strength. It's because of the pus, abscess and so on. So be careful with that. So if you're not lucky, you're going to get it. Of course, not all soil contains Pseudomonas, but in certain places, especially with animals around, then you may have it. So understand that. So when we do the virus isolation, we don't do it openly on corridor in the office room. We have a special chamber in the lab. We call it biosafety level 2, biosafety BSL2 cabinet. BSL2 cabinet. This is actually for us to handle potentially hazardous contaminated or contaminated bacteria, virus, whatever material. So we use BSL2. When we do the tissue culture, we don't use BSL2 because tissue culture is already sterile. To grow the cell, you need to grow the cell. You will learn that how to grow the cell. We don't use BSL2. We use laminar airflow cabinet, LAF. We call it LAF, not BSL, LAF. Okay. What is the difference between LAF and BSL2 chamber? So you can see that, people sit on the chair, then after that they have chamber in front. You can see, So BSL, the air flow is actually, they filter, they suck in the air from outside into the chamber, but they filter. You know why? But the LAF, laminar airflow, they blow the air from inside towards you. So the air inside is sterile to blow it to you. So you are safe because for laminar airflow to handle the cell culture, the cabinet there, they have special box there, the air will blow to you. They have filtered air inside, blow to you. It's a clean air blow to you. So can you imagine that if you handle, let's say, bacteria in laminar airflow or virus, the air blow to you? So, right. So that's why never ever use laminar airflow cabinet to do the sample, the infected sample, to process a tissue sample, whatever sputum, animal sample, human sample. You must use proper chamber. Not many people don't know this. They see that, oh, they are the same. No. Can we use, let's say, do tissue culture inside the BSL? Yes, you can, but it is not advisable. It is because the air inside, it is not as cleaner as laminar airflow. Okay. You know how important is clean air? Are you familiar with microchips or not? Computer microchips. Okay. Laminar airflow inside the one that you're going to use the lab normally, even though clean air inside the cabinet is already filtered, per liter of air every minute is already a few 100,000 particles, small particles from the environment. You stand like this right now, you're moving a bit, we are talking about a few 100,000 particle released from our skin, from hair, from my mouth, from my nose. Even I move like that, particles, fine, you don't see that. 500,000 easily. You cannot have that when you produce microchips, because now small particles go to microchips. Microchips will have electrical connection, then you get error. So microchips actually zero particles. The machinery that they have, zero particle per liter per minute. We have here about 500,000 particles per minute, per liter. We release from body. That's how now they wear kind of this space suit, special chamber to do microchips. It is not about virus here, it's microchip. See how the technology is all about. Understand, eh? So this is what we call clean room, clean air. That's what it is all about. So it is really interesting about you want to learn more. So the lab space must the lab space. What you need to do is actually to wipe out the lab with the what we call this alcohol, 30% alcohol and so on. And then before you start using it, then you need to use flame. Flame. Bunsen burner flame ataupun Bunsen burner near to you to make sure that your utensil you can burn it a bit. And then instrument utensil also auto clean and so on. And sample processing, that's something that I wish you can do, because it is even though it is simple, but it is actually a good experience for you all to do. But unfortunately, cannot do it right now. I'm not sure whether I can have a video on that. Assume that, let's say, you have liver sample, skin sample, skin sample of chicken from chicken. They suspect. They send you chicken sample, skin sample. They suspect that this chicken already affected by fowl pox virus. Fowl pox is actually a pox virus infecting chicken. Okay, not chicken pox virus. Chicken pox is actually herpes virus infecting human. Fowl pox virus is actually pox virus infecting chicken. So you want to test, You want to test whether this sample containing the virus or not. And then you want the virus, a live virus, and then you want to grow virus in your lab. So how are you going to process it? Okay. Normally, when people send sample to the virology lab, they have to tell you what kind of test that you need to do. They cannot simply fill out the form. Okay, Prof, I have a sample from there. Can you test for me what kind of thing inside? I cannot do it. I will start because there are thousands of viruses that need to be tested. Do you have money to pay me? That's the thing. So before you start to do the test, you already know what kind of what we call this what kind of test that you suspected need to do for what virus. So that is actually you need to have what we call this differential diagnosis first before you do it. Samples that need to be sent to the virology lab, first of all, when they take it from the field, when they take it from animals, they must preserve it well. First of all, advisable to use black or dark bottle, plastic bag, whatever. Second, they need to put it in the media, sterile media, we call it transport media. You cannot simply take swab and bring swab to the lab just like that. No, normally virus will die because of the environment, desiccation, very fast. Malaysia, you are lucky, you're talking about 90%, 80 to 90% humidity. But you go to places now, 66% humidity in the desert. By the time you bring it, few minutes, no, you bring the samples outside of the animal body. Most virus die already because of the desiccation. So must understand all of this. Okay. So that is how to take sample. It's not easy, just take sample and you won't, most of the time you do that, you won't get the result. You won't get the virus that you have selected. That's a problem because you don't know the technique. So when you bring the sample to the lab, you must know the media that you should use for the virus, for the sample, for taking sample from animals, the media. We use it transport media, and the media is actually very similar media we use to grow cells. When the virus, when the cell can grow, when the cell can grow in that media, virus also can grow in the cell in that for that media. That's why we use that media to transport the virus. Okay. So sample processing, you get normally when you get, let's say, you see the animal get a disease, which part of the tissue or sample that you have to take? Let's say, oh, I have, let's say, animal got liver problem, suspect virus, no? Are you going to send the whole tissue, 1 kilo of liver from the cow to the lab? Cannot. People in the field that really would like virology people to do the test, you must determine a certain which parts of the organ should be taken and put in the transport media, send it to virology lab. Must know that. You know, you cannot take the whole liver. How am I going to test the whole liver in the lab? You know, that's rubbish. You must know that. Okay. So any question about that? It's a sample processing. We need to show, but if I describe to you, you don't appreciate what I'm trying to tell you. So when you process sample, your aim is actually to get the virus from the cell, infected cell, out in the sample. You break the cell, then the virus come out. That virus you are going to harvest and do the test propagation. Okay. Okay. When you do, let's say, in virology, when people send sample to virology lab, they always put there, what kind of test you want us to do? Isolation and identification. Very common. This word actually, this terminology from postmortem. Postmortem, they have a section there. You can see the application form. What kind of test you do? Isolation and identification. If you do that, if you do that, you can say that PCR or this virus. That's easier. If you ask us to do isolation question, meaning that you want us to grow the virus in cell, and I need to identify what virus is growing in the cell. 2 steps, and it's very expensive technique, and then it is very long. Because I will ask, what kind of virus you want to test? I need to find the types of the cell that's suitable for that particular virus, because virus needs specific kind of cells. Not all virus or a virus can grow in many types of cells. So I have to predict. If it is herpes virus, chicken herpes virus, they will grow in certain mammalian cell, not all type virus. If it's actually fish virus, you cannot use chicken cell to grow fish virus. They will never grow. That's why I need to know. See? So this is about what we call this sampling and how to grow virus. Okay, so I think I need to stop too much for you today. So let's go for Q&A now. So who want to ask me questions? No? You can ask me anything about virology, maybe about other things. Okay, I can help. I will help. I just want to know, can you come to the faculty or not? Not allowed. Are you allowed to come to the faculty?
No, doctor.
No. Okay. Right.
No, prof.
Oh, sad. Okay, any question there? You need to see that, otherwise you won't appreciate.
Prof, can I ask something about COVID-19? Ya, lama nampak muka. Sorry.
It's okay, no problem, no problem. I can hear you now.
Okay, Prof. So like since COVID-19, we don't have any vaccines yet, kan? So what is actually the treatment untuk
Treatment untuk vaksin untuk ni COVID-19.
untuk COVID-19?
Okay, for virus, number one, you want, in principle, okay, virus, once you get it, okay? You cannot do much. You can only prevent. It is less on therapeutic, it's more on prophylactic. So before you get, let's say, virus, what we call this infection, you need to be vaccinated first, so that your body develop the immunity. Whenever you get infection, yes, you are protected. But if you don't have that right now, what you're doing actually, if you want to do therapeutic, meaning that you don't have immunity, you are infected by the virus, by a virus or the virus. The only thing that you can do, if you know about the what we call these mechanisms of replication of the virus, you can use antiviral chemicals. So if I'm not mistaken, there are, I think, few antiviral agent that they use to treat this. But in terms of efficacious, sorry, I'm not sure about that. You see, in the news now, there are so many things. Few months back, we can produce vaccine within 1 month. Some said, then after that, vaccine will be ready in 2 months' time. It is not like that. Vaccine, papilloma virus vaccine, like a virus vaccine, it takes 20 years to develop that until you all can use it in large amount. Maybe in 2 months or 3 months' time, that is actually experimental. But you cannot use, let's say, for a few billion people on Earth. No regulatory will allow you to say, yes, I can do that. And whatever you heard actually in the news, and you're ready, and so on, next month, all those actually wishful thinking, because for anything to be released into the market, this one commercial, must get approval from the authority, and they have to go through steps. Let me tell you steps. Step one, for any vaccine, this is law. This is not because of you can simply not to follow. It's law. So step one, you have to do the animal testing, lab animal. Then after that, large animal, 2 different species, non-monkeys. Then after that, you have to, and then how long it takes, how much money it costs you. Okay? To tell you, in principle, nothing less than 8 to 10 years to develop one vaccine for human. Papilloma virus vaccine, 20 years. Even the animal virus vaccine, it takes about 7 to 8 years before it can be produced and in large amount and the market can use it. So in human, they have clinical trial, phase 1, phase 2, phase 3, phase 1A, phase 1B, phase 2A, phase 2B, phase 3A, 3B. So how long it takes to complete phase 1? What's phase 1A? It depends on the virus, the disease. Phase 1A already needs about 30 to 40 people. Phase 2 requires 300 to 600 people. Phase 3, you need to have 6,000 people for you to test. Where are you going to get these people to test your vaccine? I ask you that question. Can you as an authority in that, can you answer that question? Okay. If anything happens to those people to be vaccinated, who's going to pay for them? Meaning that you need insurance. Who's going to pay for insurance? Millions ringgits. That's why papilloma virus vaccine, it takes $2 billion from the lab to the market. $2 billion, 20 years altogether. The cost of developing. Imagine that. Who has that kind of money to spend to develop that kind of vaccine? Because the result, you don't know. Just like dengue virus vaccine, there are now about one or two dengue viruses vaccine. There are few more coming. In the beginning, about few hundred million US dollars in 2008, 2007, 2008. I see that they just burned half a billion US dollars, no, half a billion, about 2 billion ringgit Malaysia. But they didn't get anything out of it. Who has that kind of amount of money to spend to invest for the potential long term outcome? But in principle, maybe they do. Four companies come out with the 400 million US dollars, and all of them got burned. Nothing. So you have a lot of money to spend, then you can develop a vaccine. Otherwise, I don't think we can do it. So coming back to the question, what can we do? In principle, we cannot do much because now the therapeutic this one, the virology, actually virus more on prophylactic. Therapeutic can be done, but the danger is now if you use let's say chemical, the virus tend to mutate, mutation will occur. So that is actually, and there is no there are news happening about the use of certain what we call this chemical there, but I don't know to what extent it's actually effective. Okay, I haven't read the data, I haven't seen the scientific report on that. But there are, let's say, two or three companies, and then their share price increase because of the demand for that particular product is. Yes. Any more question there?
Okay, thank you so much, Prof.
Yeah, it's not easy to answer your question because there's no material is not there yet.
It's okay.
Yeah. Some of them use quinone, to do that. I don't know the efficacy of that. Others, no questions? So my advice, the slide that I show you is a guideline for you. I hope that you do your own reading to understand further about these viruses. Okay, the basic around it. If you can do your own reading, you can see the YouTube, whatever, on steroid techniques and so on. You can, in fact, you can send me email if you don't understand about that. Don't worry about that. I'm not eating anyone. I'm not man eater. You can ask me a question. Don't worry. Yeah. Any other questions? No, can apologize because I couldn't open my webcam due to some technical issue. Okay, it's fine. Okay, if there is no questions, so our lecture will be 10 to 12 on Wednesday, right? This one, for this session, if I have an opportunity, I'll try to find material and then I share with you all. And maybe I can get my staff to dig out something from the lab about the lab sessions. Today, I need to give you this lecture introduction, otherwise you wouldn't understand what I'm talking about for the lab, Okay, right? Nothing? So, if there is no or that, If there's no other things, then okay, I wish you all the best, and I'll see you on Wednesday. Okay. Okay. Thanks. Any question? Post me. Bye. Bye.
Thank you, doctor.
Thank you. Thank you very much.
Bye.
Thank you.